DNA replication. Overall process of eukaryotic DNA replication. Let's study this process of DNA replication in detail. DNA is synthesized on the existing DNA strands of the double helix of the DNA molecule. The entire replication process is carefully controlled by a number of enzymes and other proteins: helicase, DNA polymerase I, DNA polymerase III, primase, ligase, primer, single-strand binding proteins, topoisomerase. The separation of the double helix occurs at the origin of replication. Replication of large DNA molecules may begin at several origins of replications to speed up the process. Origin of replication is a specific sequence of DNA to which proteins that initiate DNA replication bind. Helicase unwinds DNA double helix with the expense of energy as ATP. The separation occurs by breaking hydrogen bonds between the base pairs of the two strands. This is important for exposing the two strands to function as templates for new DNA synthesis. Unwound area of the double helix is known as replication fork. The parental DNA strands are used as the templates. Single-strand binding proteins bind to exposed single-stranded DNA to prevent repairing of the separated DNA strands and stabilize them. Topoisomerases are working ahead of the direction of DNA synthesis. Topoisomerase enzymes introduce breaks on one or both the strands of DNA, twist the molecule to relieve the strain and then reseal the cut ends. In order to synthesize a new DNA strand on a template, complementary deoxyribonucleotides should be added, one after the other in the correct sequence. This is done by DNA polymerase. But DNA polymerase can add nucleotides only to the 3' end of an already formed nucleic acid chain. For this, a small fragment of nucleic acid chain is sufficient, and this fragment is called a primer. Primase is a type of RNA polymerase, which can initiate synthesis of RNA on a DNA template by adding ribonucleotides. Primase adds a short RNA primer on the DNA template, forming DNA or RNA hybrid, to facilitate the action of DNA polymerase. Since the enzyme that synthesizes new DNA strand can move only in one direction, one of the new strands can be synthesized continuously, while the other will be synthesized as small fragments. The strand that is copied continuously is called the leading strand. The strand that is copied as fragments is called the lagging strand. The small fragments of lagging strand are called Okazaki fragments. They are called Okazaki fragments to honor the Japanese scientist who discovered them. Let's study the process further, by several steps. Formation of primers, by the enzyme primus. One type of DNA polymerase initiates DNA polymerization by adding deoxyribonucleotides to the 3' end of a primer. Only nitrogenous bases instead nucleotides have been used to demonstrate the elongation process here, for easy handling. Nitrogenous bases represent nucleotides, therefore, and continues polymerization on 5' to 3' direction, elongating the new DNA strand. Further unwinding of double helix, and elongation of the new strands. Completion of unwinding process, and the elongation of the strand. Another kind of DNA polymerase, known as DNA polymerase I, remove RNA fragments, and replace them by DNA. Ultimately, another enzyme called ligase seals the DNA fragments. Ligase joins newly synthesized adjacent fragments, making a phosphodiester bonds, and completes the strand. Each newly formed DNA molecules contain one parental strand and a newly made complementary strand. This replication is called semi-conservative replication. Therefore, the daughter DNA molecules are almost identical to the parent molecule, and off course to each other. Next, these newly formed linear DNA molecules are coiled to form the double helix. Function of topoisomerase. The untwisting of the strands at one place causes further twisting and strain on other place. Topoisomerase involves, to solve this problem, in DNA replication, which takes place in living cells. There are two main types of topoisomerases as topoisomerase I and topoisomerase II. Topoisomerase I makes cuts on one strand, twists the molecule to relieve the strain, and reseal the cut ends. Topoisomerase II is able to cut and rejoin double-stranded DNA. Function of topoisomerase II is somewhat complex. It allows another double-stranded DNA to pass through the cut and seal the cut ends. Hope to discuss about the function of topoisomerase in detail by another video. Proofreading. The DNA polymerase is almost 100% error-free in adding correct complementary nucleotides to a growing DNA strand. However, one error in 100,000 added nucleotides is possible. But due to proofreading mechanism, this rate will be reduced to 1 in 10 billion. When a wrong nucleotide is added to the growing chain of DNA by DNA polymerase, this mismatch is identified by the same enzyme. Then stops adding the next nucleotide, removes the incorrect nucleotide by exonuclease activity, and continues again its polymerase activity. This is known as proofreading. Thank you for watching. Subscribe for more such videos.